Welcome to AP Biology. This video is on the chromosome theory of inheritance. The chromosome theory of inheritance studies the physical basis behind Mendel's laws, or in other words, it shows how Mendel's laws are demonstrated through the process of meiosis. Mendelian genetics is very theoretical. Mendel did not know about genes, he did not know about DNA, he did not know about chromosomes. We could look at Mendelian genetics by analyzing what happens during the process of meiosis. Here we see a good example on the left. Meiosis easily demonstrates the law of segregation. The law of segregation states that during the formation of gametes, the two alleles for a trait are going to separate. Each gamete will only receive one of those two alleles. Here you see the two parents, and you see this first parent here is heterozygous, and you see that these two alleles are going to segregate when the chromosomes separate from each other during the process of meiosis 1. So during meiosis, the homologous pairs separate. When the homologous pairs separate, the two alleles for a gene separate. Each gamete is only going to receive one of those two alleles. The other parent here is homozygous, so no matter which way the alleles separate or segregate, this is the only possible combination of alleles that you can have in the gametes. When you perform a Punnett square, you are looking at all the different ways that the father's gametes can match up with the mother's gametes, or you're looking at all the different possible ways that the father's alleles can match up with the mother's alleles in the offspring. We know that each gene has a specific location on a chromosome, and that gene's location is called its locus. An exception to Mendel's law of segregation takes place when we looked at linked genes. Linked genes are when genes are associated with very specific chromosomes. An example of linkage is with sex linkage. Sex-linked genes are the genes that are found on the X chromosome. We know that males are XY and females are XX. So for a sex-linked gene, females have two copies of that gene. For a sex-linked gene, males only have one copy. Right? That gene is not found on the Y chromosome. Males are more likely to inherit a sex-linked trait because they only need one copy of that gene in order to get that trait. For a sex-linked recessive trait, females would need to be homozygous recessive in order to express that trait, and it's less likely to take place. Males inherit a sex-linked trait from their mother. This is because they inherit the X chromosome from their mother. They inherited the Y chromosome from their father. So males are going to control the sex of the offspring depending upon whether or not they pass on the X or Y chromosome. But the female, the mother, is going to control whether or not a male has a sex-linked trait. Sex linkage was first discovered by a scientist named T.H. Morgan, and he discovered sex linkage by looking at traits found in fruit flies. And in particular, he looked at this trait of having white eyes. This white eye trait is sex-linked and was found much more often in males. There are several sex-linked disorders. One of them is Duchenne's muscular dystrophy. A second one is hemophilia. Another one is color blindness. Another one is male pattern baldness. For all of these recessively inherited sex-linked disorders, males are more likely to inherit them because they only need one copy of that recessive gene, or that recessive allele I should say, and they inherit the sex-linked disorder from their mother. So what's on that Y chromosome? Right? The Y chromosome is very small, it's very reduced, it has just a few genes. One important gene is called the SRY gene, And the SRY gene is what makes a male a male. It directs the development of male anatomical features. So the X chromosome has many important genes. The Y chromosome does not. It basically just codes for maleness. We also have linked genes, and this is where we really go against Mendel's law of independent assortment. Mendel stated that every character is inherited independently. Just because you have blonde hair does not mean you're going to have blue eyes. That does not always hold true. If two genes are found on the same chromosome, they are very likely to be inherited together. Here you see on this one chromosome here, you have dominant G and dominant R. So if you inherit this chromosome, you get both big G and big R. The other set of alleles on the homologous pair here are little g and little r, so when you inherit little g, you also inherit little r. If genes are found on the same chromosome, they get inherited together. The only way that they get separated is if crossing over takes place between them. If crossing over takes place between these two genes, they will be separated. How do we know that genes are linked? You can perform a test cross. Here you have an individual who is heterozygous for two different traits, and you cross that individual with someone who is homozygous recessive for both traits. If you perform the Punnett square, the predicted ratio of offspring would be 1 to 1 to 1 to 1. So if you're looking at a number of fruit flies, you would expect an equal number of each type. Some of the offspring would retain the parental phenotypes, meaning they would look exactly like their parents. Other offspring would be considered recombinants. To be a recombinant means that you do not have the same phenotype as your parents. You are the result of genetic recombination. In this case, you're the result of independent assortment. You're the result of those chromosomes lining up a different way, and therefore you inherit different traits. When you look at the actual results in this case, the observed number of fruit flies does not match what we expected. You did not obtain that 1 to 1 to 1 ratio. Instead, more of the offspring retain the parental phenotypes. This is evidence that these two genes are linked, that they are found on the same chromosome. The small number of recombinants that you did get, these recombinants are the result of crossing over taking place between the two genes. The further apart two genes are, the more recombinants you would have. The further apart two genes are, the more likely crossing over is going to happen between them. The closer the two genes are, the less likely crossing over is going to occur between them, and the smaller number of recombinants you're going to obtain. Now, how do you know that the observed does not actually match what you expected? Sometimes it could be close. You'd have to perform chi-square analysis. If you were to figure out the percent recombination, or the percent of offspring due to crossing over, you would take the number of recombinants and divide it by the total number of flies. And for this experiment here, we would get a 17% recombination, or 17% of the fruit flies, their phenotypes were caused by crossing over. Using this information, we can create a chromosome map. A chromosome map is also called a linkage map. And the linkage map gives you the order of genes along a chromosome. In order to do this, we use recombination frequencies. It's all based upon the fact that the further apart two genes are, the more likely crossing over is to occur between them. The closer two genes are, the less likely crossing over is to occur between them. So by looking at recombination frequencies, we can analyze the distance between genes. We could tell their order on a chromosome. For instance, here, if you're looking at these three genes, B, CN, and VG, whatever they are, it's not important, you see that between B and VG, we have a 17% recombination. Between B and CN, we have 9%. Between CN and VG, we have 9.5. Now, the numbers don't work out perfectly, but obviously, since B and VG have 17% recombination, they are the furthest apart. Since B and CN have 9% and CN and VG have 9.5%, you can make an educated guess that this gene for CN here sits right smack in the middle of the other two genes. There are several disorders caused by mistakes that take place during meiosis. These disorders are usually caused by a process called nondisjunction. Nondisjunction occurs when chromosomes fail to properly separate during meiosis. Here you see nondisjunction during meiosis 1. The homologous pairs fail to separate, both go to one side. As a result, some of the gametes have too many chromosomes, some of the gametes have too little. Here you see nondisjunction occurring during meiosis 2, where the sister chromatids fail to properly separate. And this is also going to result in some gametes having too many chromosomes and some gametes having too little chromosomes. Nondisjunction causes aneuploidy. Aneuploid cells are cells that have abnormal numbers of chromosomes. And this causes many different disorders. One of them is trisomy 21, which causes Down syndrome. With trisomy 21, we have nondisjunction taking place in the 21st pair of chromosomes. If we look at this karyotype, you can see that this individual has 3 of that 21st pair, which causes Down syndrome. Here you see a picture of Klinefelter syndrome. This is caused by nondisjunction of the sex chromosomes. These individuals are XXY. Since they have the Y chromosome, they are a male. However, they do have some different characteristics. They typically have a lack of body hair. They typically have a pear shape and breast development. Finally, we have a disorder called Turner syndrome. This is X0. These individuals are female. They are also often sterile. In females, females have 2 X chromosomes. Females only actually require 1 X chromosome. The second X chromosome becomes inactivated and forms a structure called a bar body. This can create some traits in females as well, because in some females, 1 X chromosome is activated in some locations of their body, and the other X chromosome is activated in other locations of their body. This creates something that we can call tortoiseshell in cats. If you look at this cat here, you have an X chromosome that has the gene for black fur activated in some regions, and you have the X chromosome that has the gene for orange fur activated in other locations, giving this cat this patchy appearance to it that we call tortoiseshell. Mistakes can also take place during crossing over, and these could often have very harmful or even fatal effects. During crossing over, deletions and duplications would go hand in hand. When the chromosomes cross over and they exchange pieces of genetic material, sometimes both of those pieces could go to one chromosome. So that one chromosome would have a duplication that has both pieces attached to it. The other chromosome would contain a deletion. It would be missing a piece. You could also have an inversion. An inversion will be when that piece of the chromosome goes on backwards. Right? That's the least harmful of the different mistakes that could take place during crossing over. The piece just attaches to itself, attaches to the chromosome backwards. A translocation takes place when 2 non-homologous chromosomes cross over, and this can have potentially drastic effects as well. Here we see some examples. One syndrome caused by a deletion is called cri du chat, which stands for cry of the cat. This is caused by a deletion on chromosome 5. A child born with this syndrome is severely mentally impaired, and they have a cat-like cry. They usually die very early in infancy. A cancer is caused by a translocation. This cancer is called CML, chronic myelogenous leukemia, and it's caused by a translocation that takes place between chromosome 9 and chromosome 22. You can see that these two chromosomes undergo crossing over, so they exchange pieces of chromosome when they're not supposed to. This translocated chromosome number 22 is called a Philadelphia chromosome, and that is indicative of CML, a particular type of cancer. Finally, we end with an exception to the standard chromosome theory of inheritance. Sometimes it does not matter the combination of alleles that you inherit, but it matters who you inherit what allele from. Sometimes you only express the allele that comes from your mom. Sometimes you only express the allele that comes from your dad. This process, in which your combination of alleles doesn't matter, but who you inherit what allele from, is called genomic imprinting. An example is seen with the gene for insulin-like growth factor 2. We call this the IGF-2 gene, which codes for a hormone called IGF-2. And this hormone controls growth. For this trait, the only gene that gets expressed is the paternal allele, the allele that comes from the father. The maternal allele is not expressed. Here we see this example in action. So you see an individual who is heterozygous, and here we're looking at mice. The paternal allele and the paternal chromosome is shown in blue. The maternal chromosome is shown in red. Both of these mice here are heterozygotes. The mouse on the left here inherited the normal allele from its father, you see it in blue, and it inherited the mutant recessive allele from its mother. So you have the normal allele from the dad, the mutant allele from the mom. This mouse is normally sized. Its gene is properly functioning. It's a heterozygote. The mouse here on the right is also a heterozygote. It has the same combination of alleles, except this time it inherited the mutant allele from the dad and the normal allele from the mom. This mouse is a dwarf. It does not have a properly functioning gene, even though it has the same genotype as this other mouse. The only allele that is being expressed is the allele that comes from the father. We call this genomic imprinting. Alleles are silenced through a process called methylation. By adding a methyl group to the DNA, it condenses the DNA and shuts off that allele. This concludes our video.